But before we give this session to the moderator, I would like to give a brief introduction about our moderator. Our moderator today is Mr. Trihandoyo, SP PhD. The formal education, Dr. Trihandoyo was studying in undergraduate program at University of Jember for Bachelor in Agronomy and doctorate program at Osaka Prefecture University for Plant Biochemistry and Physiology. And also he had several expertise and several professional experience. And now Dr. Trihandoyo was a lecturer in Faculty of Agriculture of University of Jember, Indonesia. So ladies and gentlemen, please give applause and warm welcome to our moderator, Dr. Trihandoyo. For Dr. Trihandoyo, the floor is yours.
Thank you very much for Master of Ceremony to introduce me. I would like to introduce our guest speaker today. Is Professor Fahrul Zaman bin Huyop. Are you there, Professor?
Yeah, yes, I am. I am here.
Okay.
I'm here.
Professor Fahrul Zaman bin Huyop obtained of graduate school in biotechnology at United Kingdom and took a PhD degree in biochemistry and molecular biology also in United Kingdom. Many publications have been written, 180 scientific journal, 22 book chapter and 3 editorial member, and 4 research book. Now administration post, Deputy Dean Research and Innovation until 2017, and Assistant Dean External and Global Engagement from July 2018 to 2019, and also international contributed and reputation as a chairman for international conference as for bioscience and medical engineering 2019 in Bali, co-chairman for international conference for biosciences and medical engineering 2021 in UTM, and co-governor international conference by Biotechnology Society of Nepal 2021. And also he was chief editor of the journal and member of editorial board, chief editor the Journal of Tropical Life Science, editorial board AIP Proceeding International Conference on Biosciences and Medical Engineering, editorial board International Journal of Science and Biotechnology, to be Tech Turkey, and also Journal of Fungi Biotechnology, Cleanup Special Issue, this reputation journal, MDPI. I think time and screen for use, Professor Fahrul Zaman.
Okay, thank you very much for the brief introduction. I sincerely thank you very much to our chairman this afternoon, Dr. Trihandoyo. And of course, I would like to sincerely acknowledge my colleagues at Jember University, Dr. Tri Agus Siswoyo, for introducing me ICALS 2021, because we have been known together since we were we were friends during the previous conference in UTM, and also we have some other linkages with Brawijaya that Professor Tri Agus wife there in Brawijaya, and we had always keep in touch for ever since. So I sincerely thank you very much for the committee member for inviting me this afternoon to share my experience. I like to say the words of experience rather than my expertise, because it is difficult to define the expert area, because everything is the learning process, so that the knowledge is always been developed from time to time. So therefore, here we are to share what we try our best to share our experience and knowledge and so on. So I wanted to share my slides. Stop for a moment.
Sorry, Prof. Sahrul, maybe your speaker is still good.
All right, can you hear the voice? Sorry about that. Yeah, the topic that I would like to bring up this afternoon related to micromolecular biology method understanding pollutant degradation. So basically, what we can understand from this degradation, normally we isolate a single microorganism. Sometimes it is not very effective as what we see from the environment. So that when we bring our sample to the lab, sometimes it's not very effective. So therefore, there must be something ongoing in the natural system compared to a single organism that we talk about in the lab. So I try to match my regarding agricultural activities. So actually, maybe most of the people they talk about agriculture industries in terms of we wanted to make our to increase the yield of the food products. But my talk is more towards to clean up the agricultural based industries waste residues. So that's what my field of interest are. So if this what I call it the waste is not properly managed, so it will cause a lot of damage in terms of pollution, and it is very harmful to the human and also the animals. So what are the examples of waste? We talk about manure, oils, fertilizers, pesticide, herbicide, and so on. So these are the main concern at the moment. If you would think about what are the agriculture-based industries is all about, is about what are the waste from it. So in terms of the focus area, pesticide, herbicides, it is they are persistent. I like the word to use the word persistent. Means they are not able to be degraded properly. If this is degraded, but still there are some traces of these elements that can cause pollution. So some of the microbes, of course, there are some limitation of the microbes to do that, because some of these chemicals persistent in such a way that many microbes died because the presence of these toxic materials. So number two, apart from this toxic, in terms of the system or microbial physiology of the microbes, will not allow these chemicals to enter the cells, so that there is no expression of the gene to allow the production of enzymes specific to degrade these pollutants. So those are the three limitation I can say regarding the herbicide and pesticides. That's why if you look into the system, that sometimes these chemicals cannot be degraded due to these few reasons. And of course, if you see these chemicals, if they are not treated well in the environment, of course, it can cause cancer, Parkinson's disease, Alzheimer's, birth defect, and so on. So that's why it is very important to have this pollutant degradation. So we also use fungicide. Fungicide contain mercury, also can cause some damage to our nervous system and affect our kidney as well. So these are the examples. And then of course the fertilizers also, which is very unpleasant if it is exposed too much in the environment. Of course, it is very important to allow faster growth and increase the yield, but also it can cause aquatic environment leads to eutrophication because it will allow too much organism to live in that particular area and cause depletion to the oxygen and eutrophication occur. The treatment of environmental pollution, normally we use bioremediation technologies. These are the best, I can say cost effective for some reason, and continuous effort are required to develop the sustainable environmental microbial remediation for environmental management. So this is very important indeed at the moment. But bear in mind, the words viable but non-culturable. So as a scientist, I mean, sometimes we wanted to know what are the microbes are present whenever we bring the sample into the lab. So we must bear in mind that not all microbes are able to grow in our media in the lab. That's why we call it viable but non-culturable. So I wanted to tackle this information based on our studies in Turkey recently. We wanted to see what are the major organisms present in high salt condition and what are the majority microbes are present and how these microbes behaves. I mean, so we can predict that this microbe, the major microbe, is the one that may be important to allow pollutant degradation. And not a complete result. I mean, just something we have to further investigate in the future. So we believe that this uncultured bacteria may play an important role in the biodegradation process in the environment. So that's what we believe. If you like to say it is, we like to hypothesize this kind of idea. So as we see the lab studies versus molecular approach, so we try to match between these two things. So we like to use the molecular ecological approach. These are only the feasible methods to study the diverse of the microbes present in the polluted area itself, rather than by growing the cells, because you know the growing methods techniques in the lab sometimes will only allow culturable but will not allow unculturable due to certain circumstances that we provide in the lab. It is not suitable for this type of microbe to grow. So we must bear in mind, in the environment, we have multiple species. They share one ecological niche, and one population is the best, can adapt the best at this ecosystem. We call it, they are really predominant. So others is like minorities. So using what we call it, molecular ecology method, we can detect the dominant species, whereas using traditional methods, dependent methods that we use, isolation and so on, will not be able to detect the whole population. So these are the ideas that it is not new idea anyway, but we will use this for different model of studies. So what are the major cause that we cannot do this? Because due to the differences of physiological conditions between the lab and natural ecosystem. Of course, we understand that whenever we grow this organism in the laboratory, so normally we have differences in terms of nutrients, media for the cultivation of pollutant degrading microorganisms. In lab culture contains much more higher concentration of the target pollutant, so this will allow some microbes to grow, and also the nutrients that we have in the lab culture, which is higher. And then the microorganism utilize various cooperative interactions with other species for their growth in the ecosystem, which include syntropic interaction and cell-to-cell communication, which may not happen maybe in the natural ecosystem or in the lab, but they are so different in terms of growth condition. So therefore, there are some failures in terms of between the two systems. So what happened in the environment actually? So the network of carbon sharing bacterial consortium in doing pollutant degradation, and then very different from the lab, and the differences in the physical conditions to presume that lab strains can efficiently degrade pollutants in the environment as they do in the lab. So the molecular approach actually to detect the catabolic genes using specific PCR and so on, we can amplify anything from this mixed sample, and then we can analyze the diversity of the pollutant creating population in the environment. So it is very convenient at the moment to use molecular approach. And this is general perspective of studying the microbial community that we have at the moment, currently available. For example, using molecular approaches, it is the one that are popular at the moment, and then using isotope level, substrate level, that we can detect what are the uptake that we have to label the substrate with the radioisotope or non-radioisotope materials. They can easily monitor this substrate into the cells. And physical approach, checking the morphological kinetic analysis that one way, and then also identification of functionally important populations by means of isolation, environmentally relevant isolation strategies, and then using molecular cloning as well. And then functionally important organisms can be identified. And also you can study in terms of in situ activity, whereby we enucleate any organism that we find can degrade certain substrates or chemicals. Then we analyze in situ in terms of gene expression, in situ physiology, and so on. Pure culture studies, and also analysis of interaction with other organisms based on syntropic interaction. So these are the general perspective showing what we call it to study the microbial community. So the molecular approach basically study about amplified. We amplified from the environmental DNA sample using PCR, and then we do the sequencing analysis. Then we can check all the possible gene that we wanted to study. And example detection method, as you see, PCR, and then the rest of the techniques that are available according to the scientific experiment available. So isotope level that I already explained just now, and also analysis of in situ activities by introducing a specific microbe. Then we analyze what has been degraded and so on. And then the new molecular approaches is actually involving all the omics. So this is the focus study at the moment that I would like to share with all the students this afternoon. The omics, of course, we talk about the genomics, transcriptomics, proteomics, metabolomics, and so on. All the omics things are the new, not really new molecular approaches, has been created more than 5 years ago, but this is something that very interesting at the moment to explore. And the aims is actually to study the molecular biology techniques for studying the functional potential of indigenous microbial communities that we select. We always use water system as our environmental matrix. We can study as well in terms of the soil, air, and so on, but we use water because we use lake in high salt lake as our model. And then analysis for DNA, analysis of RNA, protein analysis, and metabolites. So these are all the important studies that you can do. So I mean, we can show in terms of diagrammatic figure. So we talk about the DNA, RNA, proteins, and metabolites. So genomics focus study of single microorganism. You can analyze the transcriptomic and proteomic and metabolic as well for the single organism. And apart from that, you can study the community using metagenomic, which is we change meta to metatranscriptome, metaproteomics, and metabolomics. So that's the only terminology, which is all the omics thing that we always hear in the current literature. And of course, I always look at this figure Metabolomes is like I'm not very I do not know too much about metabolomic. Proteomic a little bit. So metabolome always associated with what are the products, something like sugars, nucleotides. So these are all this amino acids, lipid, and so on. So this focus area, xenobiotics, available in water, lakes, and so on. So the lakes is the focus area, the important metrics that I would like to share with all of you. So basically, we wanted to study saline, hypersaline environment. So we know that in saline, it's put too much pressure to microbes. Only certain microbes can survive in saline conditions due to the fact that the water retention in the cells might go out. That cause the shrinkage of certain microbes. But this specialized organism, they can survive in a high salt due to the fact that maybe in their systems can sustain different osmotic pressure between outside the inside the cells, so that the bacteria can survive. At the same time, the bacteria can do pollutant degradation. So this is the things that we very interested to have a look. What are the potential chemicals that we talk about, always associated with halogenated compound? What is the meaning of halogenated compound? It's a compound that contains halogen group. For example, 3-chloropropylene acid, you have a chloride ion at carbon number 3. Monochloroacetate, you talk about chloride also at acetate compound. 2-CP related to chloride at carbon number 2, whether it is levo or dextro positions. And 2,3-dichloropropylene acid means the position of chloride at carbon number 2 at carbon number 3. So this another one is 2,2-dichloro. 2,2-dichloropropylene acid or 2,2-DCP is important ingredients in herbicide. So basically, the dehalogenase enzyme can cleave the carbon halogen bond at alpha position, then replaced by OH group, and then you have pyruvate as the carbon source. So the organism able to produce dehalogenase to break down 2,2-DCP becomes pyruvate. So the pyruvate will be used as the carbon source. So that's the idea of, the function of this dehalogenase. So we wanted to have a look the potential of certain organism present in high salt condition can degrade 2,2-dichloropropylene acid, and then from this hypersaline environment. So we talk about the dehalogenase is the main focus. Without dehalogenase, it is an inducible enzyme, can allow this microbe to grow in the toxic compound. 2,2-DCP is only a model of substrate. So we use this as a growth medium to allow this mixed culture from the lake to grow, and then we check the presence of pyruvic acid. Or also, sometimes we use 2,2-dichloro. We check the presence of lactic acid and the growth experiment of the mixed population, apart from study the single population. So this location of the lake puts in Turkey, it is very high concentrated with salt compared to the normal sea. So the lake 2 is salt condition 32%, with the largest salt lake. And then it is very high salt concentration compared to normal seawater 0.8% maybe in terms of salt concentration. So this is what we wanted to study about. Okay, and then the significance of research is actually to see the bacterial diversity using metagenomic approach, and then discovery the halogenase producing bacteria from hypersaline will facilitate the removal of pollutants from highly saline conditions, and of course data on genes, metabolic activities, and bioremediation capabilities of bacterium in high saline conditions. So this is what the focus area. This is the general perspective of this possible experiment that has been done related to this investigation. Based on the initial studies, so we will see that most of the organism, based on this data, we can always see what are the most abundant and what are the most least bacteria, and basically firmicutes is the most abundant from the two areas, and then followed by this Rhombacia phyla level. So using metagenomic, we can always estimate what are the most majority of the organism are present and what are the minimum abundant percentage of organism present. So we can always estimate the whole system in terms of microbial diversity. And then apart from that, the metagenomic also can identify the general perspective, the pollutant degrading enzymes which are available in the system, which is where we assume that only small percentage can be cultured and the rest may be unculturable organism. So it is very interesting to see that the presence of the pollutant degrading enzymes such as dehalogenase enzyme, haloalkane dehalogenase, and other enzymes related to allow this organism to grow at high concentration of salt. So you see that the enzymes that are important, glutamate synthase, and then also something like actoin, L-actoin synthase. So these enzymes allow betaine aldehyde and so on, allow this organism to survive in terms of high salt concentration. And then apart from that, it is difficult because we wanted to study a single organism. So therefore, we have to forget about unculturable techniques. So we just use normal isolation of bacteria. Using high salt condition, we managed to isolate a single organism and we study the whole perspective, the whole profile of bacteria that we isolated based on two strains actually can grow in high concentration of salt plus the presence of 2,2-dichloropropanic acid as a carbon source. Then we managed to identify this organism based on CCS RNA analysis to determine the species and genus name. And also we proved that this organism can grow in the presence of 2,2-dichloropropanic acid and with cell doubling time of 8 hours compared, and also the release of chloride ion as well. And then we check in terms of chlorides and also the pH, and then the temperature, the optimum temperature, and so on. I don't have to deliver all this data. And then we can check that we can amplify the dehalogenase gene, and then we estimate the size of the gene, and then we characterize the proteins based on in silico investigation. So it is, so in terms of in silico studies, we have to check the number of acid amino and the stability of these proteins in the presence of salts and so on, the binding energies of the substrate and so on, in order to make sure that these are the enzymes that we believe important to do degradation, especially in the presence of high salt. And of course, whenever we do the structural functions, we have to check. We have to validate the data based on several techniques, like verified 3D methods and check and so on. And then we also check on something like what you call it, protein ligand complex. This is something like to see how the substrate enzyme bind together, to see what are the possible score that is it true or not that this organism specific substrate. I mean, based on this data, we can hypothesize. We believe that type 1 is more can react with the L position of halide compared to others like D. Of course, there's some, but not as strong binding as an L to CP. So I mean, this is in silico investigation. We don't prove that basically based on the enzyme studies. So these are the important what I call it acid amino residues, important to bind with the chemicals like L2CP just now. And then we check this in the presence of salt to see whether it is good or not in terms of the complex enzyme ligand investigation studies just now. And then using RMSF again to see the stability of the binding residues in between these two ligands and enzyme and the ligands. Just skip that one. So I mean, there are many techniques. I'm not really expert in GROMAX. Basically, we do collaborate with Islamic universities. We have expert to interpret all this data based on in silico studies. Our group in UTM, we study about this basic microbiology, growth experiment, and metagenomic analysis also done by Islamic University in Kuantan, Pahang. This MD simulation, is to check, is to verify again what are the best substrate to be consumed by bacteria based on the enzyme studies. So DEH is actually, we name the enzyme as DEH, dehydrogenase. The best substrate is L2CP compared to others, which is less effective. So we always look at this the affinity, binding affinity. The higher is the one that we assume that this best substrate. This also we have been discussed this regarding MD simulation, the binding energy as well, and then yeah. Apart from that, we study also about this the complete genome. We managed to sequence the whole genome of the bacteria. So the whole genome is like a very quite not too small, not too big. 6.4 6 million 412 base pair, and then it has a one chromosome and a number of plasmids. It is common for this dehydrogenase producing bacteria or common bacteria that can degrade pollutants. It has plasmid is 10. So in terms of the naming of bacteria based on 60S, which is slightly differ from what when we study the whole genome of, we estimate the bacteria that it is belongs to the genus of Bacillus species. And then initially we identify bacteria based on 60S, which is Bacillus thuringiensis. But once we do the full genome sequence, the bacteria now becomes Bacillus megatherium. I mean, there's this discrepancies between the two data that we have at the moment. And then what is the most interesting? We study, I mean, this is what we expected actually. Once we study about this, the whole genome studies, we find that based on simple investigation. So we look at maybe we only have 1 or 2 dehydrogenases, but actually based on this genome investigation, we found that there are actually 9 dehydrogenases which contribute to this what do you call it, to this degradation. And then apart from that, we have other dehydrogenases like 2-haloalkanonic dehydrogenase, which is present at circular chromosome, and some available at the plasmid. 2-haloalkanonic acid dehydrogenase also present at the plasmid. So these are all based on the data that we obtained from the full genome sequence. And from the genome sequence as well, analyze of these are when we study the single organism, we use PCR. So we find DH2 is the possible enzyme that is can react with L2CP. It's actually when we do the whole genome sequence, we found this similar to C6. So C6 with more or less same molecular weight. What we found from the PCR and the actual size of the gene based on the full genome. So we believe that the techniques of PCR is quite accurate when we compare to more or less accurate with the full genome sequence. So therefore we can conclude that this bacteria is actually contain more than one. So in Rhizobium system we have only 3 dehydrogenases. So we have 2 stereospecific and 1 non-stereospecific, but using metagenomic genomic studies especially. So we found that it is actually more than 3 dehydrogenases based on the model we have in Rhizobium. So that it is very, some of the genes, what we believe, some of the genes, they are silent, they are cryptics, so not being expressed. So only some certain selective genes might be important for this degradation. So it is very difficult to identify which gene will be expressed because dehydrogenase is inducible enzyme. Inducible enzyme means only certain certain condition, this enzyme will be produced. But what is the message that we learn from this whole genome sequence? That there are actually more than 3. So in terms of a single organism, we study about gene expression. We find 3 in dehydrogenases in Rhizobium species. And then if we don't make a full sequence, we manage to see only 1 being expressed using primers that we have. The rest that we cannot detect at all. So it's like, so these are dehydrogenases that more than 1. So maybe other may have special characteristics that not been explored before. So these are the novelty of the techniques that we saw using the whole genome. There are many things can be done. There's so much information that we can obtain from this full genome sequence. So not only the dehydrogenase processing bacteria, enzyme that we managed to study. So there are also important enzymes to degrade other pollutants as well. So that a single organism contain vast information like can degrade pollutant benzoate, naphthalene, anthracene, dichlorobenzene, and so on. So that once we put this full genome sequence in database, so anybody interested to study about the benzoate, naphthalene, degradation, and so on, they can always pick one sequence of the gene, then you can study about the properties of the enzyme based on the silico and so on. So this open more to academics investigation for this new PhDs, new MSc student to explore, just analyze the protein sequence and do in silico investigation. Then you can get many data. Then you can publish your work as much as you can just by doing the full genome sequence. So apart from that, another gene important, a gene that allow this organism to survive in high salt conditions. So we have this adaptation enzyme, we call it, trihalose operon glutamate synthase, choline transporter, betaine aldehyde dehydrogenase. So some of them are important in terms of uptake system. So this organism to bring in any carbon source into the cells. So some of this gene important, something like to allow the organism to survive in high salt conditions. So these are the genes which are present. What we expected to see, there must be some genes involved in terms of to make sure that organism can survive in the high salt condition. So if the gene is not present, so maybe we have a wrong isolation. We isolate something else, a contamination. But what we expected to see in this particular enzyme adaptation, we found what we are looking at. So there's no argue in terms of the authenticity of the organism that we have been studying so far. So the conclusion, the study successfully profiled the bacteria population and the abundant presence in the lake, as well as protein function that provide information related to adaptation of bioremediation potential. And the bacteria strains were successfully isolated, identified, and the ability to degrade pollutants were confirmed. The dehydrogenase gene could only be amplified in one of the bacteria, Trihandoyo. But of course, Trihandoyo name species, so we changed to Megatherium because after knowing the full genome sequence, that it is not actually Trihandoyo, it is there is more data. I don't have with me at the moment, so we are in the process of writing papers based on once we have this full genome. Suddenly we change the name of the species from Trihandoyo into Megatherium. Mechanism amplified dehydrogenase enzyme action in silico investigation in respect salt and pollutant degradation were also investigated. The mechanism, the genomic analysis of Trihandoyo reclassified it as Megatherium. This what and this what we find that between the two methods. Of course, we believe that full genome can give more data to allow the naming of the genus, species, and so on. So last but not least, I would like to thank our friends that contribute the work. We took sample from Dr. Yilmaz Kaya in Turkey, so I was there in Turkey for 6 months in his lab, and also Habibat now completed her PhD in UTM, doing all this work direct from isolation, metagenomic studies, and Roswanira involved in chemical investigation. Faraj is the one who obtained a sample and do some purification of the water and so on. And then Asmir from IUM, so he's the one who helped me a lot in terms of in silico investigation. This is beyond my knowledge. All this in silico investigation to study protein structure and functions. Too complicated. I mean, another one is Dr. Azim Naim. So he's the one who do experiment on something like metagenomic investigation. Again, metagenomic is like you have to expert in computing to gather all the data and then do some analysis. And of course, Nurul Hidayah is Dr. Nurul Hidayah help us a lot in terms of genomic investigation. This needs a lot of what you call it computing studies. That which is again because the technology always been developed, so that's why we need collaborators. The whole places, collaborators to help to make sure that the study becomes success. And Batul, Batul focus something to help us to study the degradation process. But apart from that, Batul did work on high alkalinity. Also from Turkey, also the sample we took from Turkey to study the dehalogenase properties based on growth in high alkaline. So I think that's all I can share with all the fellow students at Jember University, and also I sincerely thanks all colleagues, especially Dr. Tri Agus Siswoyo, who wanted me to share, I mean, what I've been doing for the past 2 or 3 years. So here I am. So I just deliver what we have been studied. So it is nice actually to be here. So at least I can share what has been the progress so far in our research. So thank you very much. So I pass back to Dr. Trihandoyo for Q&A and so on. Anything that I can afford to answer. Otherwise, I will reply to email and so on. Thank you very much.
Thank you very much for your nice presentation. Now we are going to answer question session. Please, audience, if you have any questions. One question from Wahyu Rizky, University of Jember. You explained that the halogenous bacteria could be remove pollutant from the environment. That means we can get a pure gold from the mining gold factory. Waste.
Yeah. All right. So thank you very much for the question. Actually, gene mining is different set of studies. Actually, I'm not very familiar with what you call it purification of gold using microbes. So there are people interested in doing so. So maybe they can remove all the traces. They can get a pure gold by the presence of this bacteria. So it is actually beyond my expertise to explain about this obtained from the pure gold. I'm sorry, I cannot deliver. I cannot explain much on gold mining. Actually, I'm very sorry about that.
But maybe it's possible to construct a new the halogenous bacteria specific to add to specific mineral.
Yeah, actually, this dehalogenase means the enzyme can react with carbon halogen. So that's the main criteria. So anything that any chemicals related, it has a halogen group 7. So this dehalogenase can react, can cleave carbon halogen group. So that's the most this is the function of this organism. So then apart from that, our model always related to what you call it aliphatic acid. Aliphatic acid means straight chain carbon. So maybe this enzyme is not function, if we have more than 3 or 4 carbon, it is not as effective function. So it is very specific enzymes. So that's why based on our genomic full genome investigation, you can see a lot of enzymes, genes that might be useful. This organism not only important for dehalogenase, but maybe for other function as well. So all I can say that this organism is rich in terms of the genes, and maybe we can use that, for gold mining and so on, to make it pure, to remove all other traces. Then we can get pure gold from this. I'm not sure. Maybe the same organism can be used, based there are many genes involved. But what we focus area at the moment is dehalogenases, and 2,2-dichloro is the model of the substrate to induce this enzyme.
Okay, thank you. For next question, maybe from audience, please. I think this topic is very interesting in the future to remediate and our environment.
Yes. But unfortunately, we never make it commercial. I mean, to improve the system, we always use this as model for too much academics, I can say. So, well, if people interested for commercialization and so on, maybe it might be a way of doing it. But at the moment, we are more towards academics rather than application to see how this enzyme react in situ. But well, anyway, in terms of biodegradation is like we always rely on in situ organism to do the job rather than introducing our own microbes to cure the environmental system.
Maybe also my question, what kind of product from the dehalogenase bacteria, if also this it also useful for us or made new pollutant?
To make new pollutant?
Yeah, I mean, when we construct new bacteria by using genetic engineering and other method, what's kind of the product of this bacteria?
Okay.
It's useful
So,
for us, or maybe became new pollutant and difficult to control.
Yeah. So this when we study dehalogenase, it is very specific, actually, very specific, very precise. So this we use the enzyme to react with selected substrate, like 2,2-dichloropropionic acid and DL2-chloropropionic acid, and also we can react with like 2,3-dichloropropionic acid. Also we can react with monochloroacetate, trichloroacetate, and dichloroacetate, and bromide, something like dibromoacetate, tribromoacetate, and so on. So the product of dehalogenation, we call it. So for like DL2-chloro, so the product of dehalogenation. So pyruvate is the simple, substrate, like for this TCA cycle. So the product of dehalogenation of 2,2-dichloro is pyruvate. So the pyruvate is actually the carbon source for the bacteria. So only bacteria produce dehalogenase can transform 2,2-dichloro becomes pyruvate. Same as if we use DL2-chloropropionic acid, we have lactate. So lactate is the product from DL to chloropropanic, because dehalogenase from this bacteria can convert the substrate of DL to chloro become lactate. So this lactate will be used as carbon source. Same as if you use MCA, monochloroacetate. So the product is glycolate. So glycolate is the carbon source as well for the bacteria. So that it is very, all I can say, very simple model, very simple model, just for the sake of studying dehalogenase using this kind of model. Yeah, but maybe if we use other substrate, so maybe it will transform to much more complex product of dehydrogenation, but we unfortunately we don't test that. So maybe, so that's why for pollutant degradation involve sometimes in the one population involve many microorganisms. If you have other substrate that I said just now, other toxic compound, so this bacteria may co-metabolism, co-metabolism. So whereas from one substrate become another substrate which is very toxic, and then another microbe utilize this toxic substrate and convert that into non-toxic. So these are all these what we call it co-metabolism might involve in the actual environment system. But what we have now is very simple model to show this dehydrogenase viable importance for the specific studies. So yes, I agree with Professor Dr. Trihandoyo just now. Yeah, it might if you use different substrate, so maybe it has different product of transformation that might might become much more toxic compared to the initial substrate. Yeah, so that's I hope that will answer the question just now.
Yeah,
Thank you.
because this topic seems like made us impossible to possible. And last question again, sorry.
Yeah, it's okay.
How long did you do this research from zero to get the new remediate bacteria?
Yeah. Isolation process is not that difficult. To study a single organism, it's not that difficult actually. We have to test a growth experiment, but that's not really difficult. But once we go to in silico investigation also will depend entirely on the PCR technique. We fish out the important band using specific primers, because using primers we can always amplify a specific gene present, and then of course we talk about study of metagenomic and genomic. So I mean the hindrance of genomic and metagenomic, all I can say that the computing, because we need people who can assist in terms of to interpret. You talk about genomic, for example. So genomic, we sequence genes in partial. Then you have to do gene assembly. There's another techniques that I don't have any knowledge on gene assembly. Then followed by gene annotation. So we need an expert. To find an expert, sometimes this work is quite complicated. We need to reanalyze again and again. Sometimes that's take longer time than what we anticipate, and also doing the metagenomic also is quite confusing. That takes some time as well. So the whole lot, I think 2 years is the time frame that you can do things completely. So it's like the growth experiment always possible, but once come to computing, because we don't really understand the language of the computing to help us more. So and then number three is the machine. The computing must have very powerful machine to analyze, like to study the structure function. So you need to have to run a big what do you call it, a big software. Of course, GROMAC you can get free of charge, but sometimes you need to have some a good computing that can help. So sometimes that facility is not available in UTM. We need to purchase a new machine. Sometimes the machine broke down as well. So that's come into factor that we have to delay our process. So 2 years is the most. But if you have all the facilities, I mean, 12 months is appropriate. So collaboration is very important. So that's why we involve many people. Sometimes my colleagues said that how come a single PhD you have so many co-supervisors? I said I can't afford to do everything on my own. So we have our colleagues to to contribute as well to make this work successful.
Okay, thanks. It's possible this gene, the halogenase genes, insert to other bacteria, seems like cyanobacteria.
To put into another bacteria, what we have been doing, we put that in the plant, we use tobacco. We wanted to see how the gene function because how the genes function in different organism. We use tobacco. The reason is, I mean, kononnya we wanted to make herbicide resistant gene. It's like we have the halogenase from rhizobium. We put in the halogenase D, for example, specific, and then we put in another gene that non-soil specific. The halogenase 2 gene, the halogenase E gene from rhizobium, we put into tobacco plants, and we saw that once we spray this tobacco using plant tissue culture techniques, there's some resistance towards certain concentration of herbicide of 2,2-dichloro. If put in too much 2,2-dichloropropanilic acid, exposed, of course, the cell will die. But certain we have some cut-off point, certain concentration of 2,2-dichloropropanilic, still the tobacco can resist. So we have been trying this. But of course, again, as I said, 2,2-dichloro or dalapon, it's not the popular herbicide in agriculture, I think. So you have other chemicals which is more, something more non-specific target of better herbicide. But the halogenase is one to effective towards dalapon actually to make plant resistant and things like that.
Thank you very much for a great answer. Thank you very much again, and
Welcome.
I hope your experience will give us increase the capacity acknowledgement for our lecturer and student. Thank you very much, Prof. Fahrul Zaman bin Huyop.
Welcome. Yeah.
And see you again maybe in Malaysia.
Yeah, sure. By all means, I always welcome colleagues from our neighbor. Yeah.
Okay.
Thank you very much.
Thank you. For next presenter, I would like invite Muhammad Rondi, MPPhD. He graduated in Socioeconomic Agriculture, University at Jember, took Master in Economic Agriculture at Gajah Mada University, and took PhD in Biosystem Sustainability in University of Hokkaido. Publication in Scopus Index Q1 and 7 article in Q2, 2 conference article, and also book Kelembagaan Irigasi Pendekatan Model Kontrak 2018. Now, Pak Rondi, as a Secretary of Institute of Research and Community Service, 2021 until now, a Head of Study Program Agribusiness Faculty of Agriculture, University of Jember, and also Editor-in-Chief of Journal Sosial Ekonomi Pertanian. To Pak Muhammad Rondi.
Yes, I'm here.
Time and screen are yours.
Is my voice clear over there?
Yes, clear.
Okay, moderator. Is my voice clear over there?
Yes, clear.
Okay, so may I start now? Okay, thank you, everybody. Let me start with Assalamualaikum warahmatullahi wabarakatuh. Thank you for Dr. Trihandoyo who introduced me previously, and then also thank you for the committee who invite me for being a speaker in this conference. Also for all guest speaker or keynote speaker who attend here, also for all participants who participate in this conference. Good day for everybody. Good afternoon. So let me start my presentation. Let me share my screen. Is my screen seen over there? Hmm?
Okay.
It's okay?
Yes, your screen is okay. Your slide is very good.
So thank you. Let me start for me to present our topic in this afternoon. So the title is the Strengthening Agriculture Institution to Increase Farmer Access to Market. This is how I start this presentation. The presentation comes from our previous research, especially for institution. So why I start with this presentation? Why I take this presentation, this topic? Because nowadays everybody know that the farmer, especially in tropical country, so they have some maybe handicap or some problem, especially how to increase their income. So let me start with the condition. This is the outline. So let me start with the problem of smallholder farmer, and then how the agriculture institutional model, and then the problem of agriculture institution in Indonesia. So in the end of this session, let me explain about the agriculture corporation, and then finally I have maybe we have some closed loop supply chain. Let me start with the main problem of agriculture in Indonesia. The first problem of agribusiness in Indonesia is the land ownership is in small scale farmer. As we know that the average of land ownership is less than 0.5 hectare. This make the farming is not efficient. We already know this. And then the second one is about the land fragmentation. Even the land ownership is less than 0.5 hectare, but the land itself is not in one area. Maybe in a small parcel in another area. So also with this land fragmentation, we have such a problem. So the small scale is not so efficient. And then because of macroeconomic condition, so the price of land is so very high. So in some business, in some agriculture business, this is not so benefit. So there is no advantage for doing agriculture, especially for food crops. Yeah, we can imagine that maybe 0.5 hectare. So the total revenue is about the 12.5 million, and then subtracted by the cost, so maybe it's about the 6 million income. So for 6 million, I think to fulfill the basic need is not so, it cannot to fulfill the basic need. So therefore, the increasing land value, it effect to the lower income for farmer. And then the second problem is agroecosystem and its supporting. So what is? So because of climate change, so nowadays we can find a lot of new pests and disease that it disappear. It would not appear in the previous, but now it's appear in these days. And then the next problem is about the limitation of water irrigation. So because of some let's say deforestation, so we have small water resources. So we can see in some mountain area, so the plant is not appear, and then in the long term it will affect to the water availability. So with the small water availability, so it has big problem. I mean, in the as we can see in the irrigation system, there is upper area or intake area. The water is enough, but in the tail area, the water is not so enough, especially for dry season. In the rainy season, the water is enough, but in the dry season, the water availability is not enough. So therefore, the cropping pattern for 1 year is only 2 times. Let's say paddy and another crop. So because of this, so in one area, it can be cultivated only for 2 times. And then another problem is not applied good agricultural practices. This is especially for old farmer. For young farmer, they can easily find the information, and then they can easily to find the market, and then they can ask how to cultivate the new crop or the new varieties in order to get the high value crops. But for old farmer, they have difficulties to do so. And then the third problem is institutional problem. So what the institutional problem? Actually, the problem itself is how to distribute the raw material, I mean the farm input, and then how to distribute to the plot, and then after that, how to get the yield to the market. Who will guarantee that the crop that cultivate it will be guaranteed in the market? This is the difficult thing. So especially farmer, they cultivate that they usually they are customized to cultivate. So the farmer itself, they don't try to cultivate what the market needs. So therefore, this is the problem here. And then the second problem for institutional problem is quality variation among small producer. For smallholder farmer, they because they do not apply the good agricultural practices, so the quality itself is vary among themselves. So therefore, the market they cannot receive all what they produce. And then the third one is low bargaining position. So in small parcel of plot, so when they also cultivate in the different crops, so the position itself is not so high. So I mean it's not balance between producer and then integrator or the market. And then for marketing problem, this one due to the climate change or seasonal condition. So planting timeline does not meet with market need. So some maybe in some area they try to cultivate another crop the market need, but because of seasonal condition, so the crop itself is not harvested well. And then the other one is no market assurance. There is no guarantee when the crop is failed and when the crop is not cannot be harvested well. And then for the business process, so not fair supply chain management among stakeholders. This is the important thing, because actually the business is the business itself. So there is no the agreement is fairness, because when they doing business with not fairness, so maybe now they can do it together, but in the next period, in the next session, so when there is no fair gain, so maybe the relation it will be break out. This is the institution, the concept of institution. So institution is different to organization. Organization is the physical entity, but the institution is the rule of the game. How to rule the organization. Organization is we know it is like the body of institution, but the institution is, itself is how how the rule to run the organization. So because of this, so maybe as we know in the previous time, so the market itself is just how to sell from a farmer to middleman and then to big market, and then after that to small market, and then to consumer. But because of recent condition, because of technology, because of information, and then the condition, the market itself is also changing. The important thing of the institution is all they do, I mean the parties in the organization or parties in the institution, they do it by incentive. When they get incentive, so they can run it again and again. But when there is no incentive, so the business will stop here. And then the another important one is religion is the highest institution. I mean the highest institution it's a little bit difficult to change religion in the short period. But for business, the period is very short, and then subject to change easily when there is no benefit anymore, so the business will break out. This is how I think we already know this one, but what the main point here is from grower to consumer. So in between the grower and consumer, there is integrator, and then how to distribute from grower to integrator. So we know three ways to distribute yield from grower to integrator or to consumer. First one is spot market, and then the second one is contract farming, and then the third one is vertical integration. So maybe we need to keep in mind that some grower or some farmer or some producer, they will try to select what kind of organization or what kind of institution they will choose. Because maybe some grower, they prefer to choose spot market than contract farming, and another grower they will or they prefer to select spot market than vertical integration or contract farming. So spot the difference between the three spot market is there is no agreement before. So when they do farming, so they do it, and after that they will try to find the market. On the market, it's not decided before. So producing good with no market connection in advance. This is for spot market. And then for contract farming, so agreement before production. So what the kind of agreement? So like quality, and then quantity, and then the price. This is the important thing. What quality the market need? What the quantity the market need? And then what the price? And then this is the difficult thing. Even though already contracted, I mean the grower and the consumer, or grower and integrator, but the two parties, they can cheat each other. Like when the quality is not good, so the price will be lower. When so how to measure the quality is also difficult thing. Let's say farmer, they have opinion that they good. I mean the harvested good is good quality, but when they bring it to the integrator, when they bring it to the consumer, the integrator will say, "Oh, the good is not, the quality is not good. So I will discount you. I will discount the price." So therefore, the two quality price is something interchanging. And then the last one is vertical integration. So chain in production to market in big related company. So I mean, in one company, they have the, let's say, core business, and then they have another business. So in one big company, like a feed mill company, so also they have a poultry company, something like this. So which one the best one? So it's difficult to say spot market is better than contract farming, or contract farming is better than spot market, and also vertical integration is better than contract farming. It's difficult. We do some research. So in one area, they say spot market is better than contract farming. So therefore, they prefer to use or to apply spot market than contract farming. In some another area, they prefer contract farming. It's different. It's depend on the grower. It's depend on the integrator also. Maybe this relation is interesting to be to be what's it to be now how they do it in some area in the world also. Some they prefer contract farming, and some they prefer spot market. This is actually let me explain what the benefit of contract farming. Yeah, the benefit of contract farming, they can transfer the risk from one party to another party. Let's say, let me give some example. So for poultry business, and also the grower, they have relation, they have contract with the feed mill company. So by doing contract, so the feed mill company, they can transfer the risk because they sell the feed mill, so they sell the feed to farmer. So actually their business is feed mill, not poultry itself. But how to manage their product is accepted by farmer, they do contract farming. Also for farmer or grower, they have benefit when doing farmer. So because sorry, when doing contract. So I mean when they do the contract, so the assurance of farm input they get. So for day old chicken, so I mean DOC, yeah, the small chicken, the company they will they will guarantee that the DOC is good, and also they don't need to pay in advance. So by doing contract farming, the grower they have incentive to do contract. This is, we call it risk shifting and then risk sharing. Risk shifting is shift from one party to another party. Risk sharing, they can share the risk itself. I mean, when the farmer failed, who will have the impact of the failed? So how many percent for farmer, how many percent for feed mill company, something like this. The second one is lower transportation cost and potentially reduce transaction cost. I mean, the contract farming, they have benefit because they already have the contract, so there is no more cost how to find the market. And then by doing so, the transportation cost is lower. And then the third one is access to credit. So farmer, they will get credit. Of course, there is interest rate in the credit because there is no, there is no freelance. So even though farmer, they receive the farm production, but the farm production price is higher than the market. When they buy it in the market, the price it will be lower. But when they buy it in the integrator, the price is little bit higher because from company point of view, they also to provide the farm input, they also have credit. I mean the credit from commercial bank. And then the third one is quality and quantity assurance. By doing contract, so the company they will receive the quality, yeah, because in the contract it's written that the quality is something already decided there. So maybe the size of chicken and then the size of fruit already written there, and also the quantity. I mean, for one season, so please provide some quantity. If not, so maybe the company, they will stop the farmer or the grower. But based on our research, there is some problem in doing contract farming, and therefore by this problem, so some farmer or some integrator, they prefer to to uh, to do not do the contract farming. Let me explain about the horticulture business. So the problem in horticulture business, misperception in quality. We have did the research in some area in East Java, especially for a potato business. So the opinion of farmer is different with the company. Even the contract is already decided. The contract is already agreed by two parties, but still the quality perception is different. Because of quality perception is different, so the price is also different. Let's say for one size, so I mean the bigger size, the price, let's say for $1, and then because of the size is not so good, and then the quality is not so good, so maybe the price is not $1, maybe less than $1. So this is the main problem in the contract. And then the second one is discontinued quantity due to weather factors. In the contract, it describes that the quantity, let's say for several tons, let's say put quantity 10 tons for one season. But because of weather condition, so the quantity is less than 10 tons. Therefore, so maybe they will reduce the price. Also, some they will stop the contract. And then this is the common problem. Some farmers sell some of yield to another market because of price in the market is higher than in the contract. Usually, it's like that. The price in the contract is lower than in the market. So therefore, the grower, they try to sell in another market. So because of that, so the contract, it will break out. The second one in the plantation crop is sugarcane business. Let's say the problem is variety of quality. It's a little bit the same with the horticulture, but the definition of quality is different. In sugarcane business, yeah, the quality described as sweet, clean, and fresh. Sweet, clean, and fresh. Because the sugar itself, it has to be transferred to the sugar, so the sugar content itself must be higher, and also clean. When there is some, let's say, maybe some something put in the truck when the farmer deliver or distribute to the factory, when the sugar is not clean, so maybe in the factory they will discount it, and also fresh. Yeah? I mean, when the sugar harvested, and the time also it will be delivered to the factory. When the time is longer than the desired time, so also there is some discount over there. Also, this one, some farmers sell zeal to another factory. It's the same with the horticulture business. And then for the poultry business, yeah, this is the big business in Indonesia because the consumption of poultry, I mean the chicken, is increase because of increasing of income. So this is a big business in Indonesia. What the problem in the poultry business? Put and put any day old chicken to poultry pen to get fit. I mean, the grower, they try to put DOC to the pen, and then why farmer doing such thing? So because put another DOC, they will get free feed. So when they when the DOC grow, so maybe the grow is not sufficient enough. I mean, it's not optimum. Usually for 30 days, 30 to 35 days, so the body weight of the chicken is 1.2 or 1.5 kg, but because of putting DOC into the pan, so maybe the weight is not the optimum one. And then the second one is selling the feed to the other grower to get cash. I mean, when they get a feed from the feed mill factory or feed mill company, so they try to sell it to another grower. Why? So because they need cash, something like this. So the third one, some farmers sell to other integrators. I mean, the selling yield or selling poultry or chicken to the another integrator because of higher price, or even to the fresh or wet market. So how to make the relation is can be continued for time to time. So this is grower and then this is integrator. So as we know in the supply chain management, so we have three point. The important thing is the flow of product, and then the flow of information, and then the flow of finance. When the grower and the integrator, they can keep this three important thing, so the contract, it will be continued. So how about the product flow? This is quality assurance. How to produce in good or in the desired quality, and then how to produce in the quantity, and then also for time assurance. Also information flow, yeah, the price, the quality assessment and price, and then the finance is credit term, and then payment schedule. This is also important, the payment schedule. Okay. This is actually how to solve how about the land fragmentation by doing or by engage in the farmer corporation. So when the land management for smallholder farmer is important because of the land ownership is less than 0.5 hectare. So land management is one of solution. And how to increase the farm scale, and then how to boost the efficiency. So the benefit of farm corporation is efficient on agriculture input use, and then effective on agriculture support, irrigation, pest and disease control, and then how to get credit, how to get easier access to the credit, because when the land is aggregate in one corporation, so maybe they can easily to get the credit. And then also it can be raise the increase the bargaining power to the market when they sell it the product in the higher quantity, so they have a higher bargaining position. But we have to take care about some problem is happening in the farmer corporation because the farmer corporation is based on the small farm land. So some problem is because the corporation become common pool resources, which potentially rising free rider when they run the farmer corporation. So who will be the leader? Who will run the business? Therefore, we have to make sure that this corporation is running well organized well. This is an alternative. Let me up to what time, moderator, for me? Is it the time is still enough? Okay. Okay. Let me continue. It's okay, moderator, for me to continue. Okay. Actually, I can hear Pak Trihandoyo voice, but it's okay. Let me check in the chat, maybe.
5 minutes
Oh.
remain, Pak Rondi.
Okay. Let me continue. This one is an alternative, closed loop supply chain management. What does it mean, closed loop? Closed loop mean how to integrate farmer to off-taker. How to integrate farmer to off-taker. Then who the parties engage in this business. So this is actually something like a new concept, how to integrate farmer to off-taker. So Minister of Finance, Minister of Finance in our ministry, so they run something like this, closed loop supply chain management. I mean, the organizer will organize how to provide the finance and insurance, how to provide agriculture input, and who provide the agriculture input. Sorry, maybe my voice something. My presentation. Who will provide the sustainable agriculture, and then who facilitated, and then who the offer. So by this such business, this is still a trial business in some area in Bandung, and then some east of Indonesia. The government still running this program. The program is success well because it's already run 1 or 2 years before. So there is some case the program is running well, and then the other case the program is failed. But still, yeah, this program we have to run such program. I mean, there is several ways how to find how to solve the problem. And then this in the closing or the summary. Due to land ownership limitation, farmers need to find high value crops hand in hand with the food security crops. And then the second one, contract farming is a way to help closely among grower and integrator. The important thing is trust among parties is the key to find fairness of contract farming. And then the third one, contract farming is a way in land management to increase farm efficiency, corporate farming. And then the last one, closed loop supply chain management is an alternative to help farmer to consumer in increasing agriculture efficiency. Thank you very much for your attention. So let we discuss further. Thank you very much.
Thank you very much Pak Rondi, for this such as informative and interesting presentation. Now we have a short time for question and answer session. Okay, we have first one question from La Sinaini from Sekolah Tinggi Pertanian Una. Why farm institutions are not able to solve farmers' problem, especially the welfare of small farmers, and how farm institution model is able to solve farmer problem starting from upstream to downstream?
Okay, thank you for the nice question from Ibu or Bapak Lasina ini. Maybe Ibu from Sekolah Tinggi Pertanian. Why farm institution are not
No. I am sure.
Okay, thank you. Why farm institutions are not able to solve farmers' problem, especially the welfare of small farmer, and how farm institutional model is able to solve farmer problem starting from upstream to downstream. Okay, thank you for the question. As we already search for our research, in Indonesia, the farm institution, let's say for contract farming, the percentage of farmer who engage in contract is very, very low. In poultry business, the highest one is in poultry business. The poultry business, the contract farmer is 55%. And then there another 45%, they do not do contract farming, or they do not engage in contract farming. In plantation crop, the percentage of farmer who engage in contract farming is very, very low. For rubber farmer, the contract farming is very low. It's still 8%. And also for the smallest one is in the food crops. In the food crop, the percentage is only maybe 1%. Why something like this? Actually, the contract farming, the benefit of contract farming, I already shared before. So the contract farming itself, they have a big benefit, even for farmer to integrator. But why they don't do engage in this one? So because of the problem of contract farming itself. Some farmer they do cheating, and then some integrator they also try to make the relation is not so fair. So therefore, the contract farming they will break out in the next season or in the next period. This is the main problem. And then how to solve like this one? In economic, we have such what would say this condition is like game condition. So I mean cheat and chat. Yeah, I mean when the farmer doing so, and then the integrator will doing another, and then when they do contract, so actually the aim of the objective of contract is to increase the productivity. But because of the problem, I mean the contract problem, some farmer they cheat and also the integrator also cheat. So maybe the contract it will break out. And then how to solve this one? They can put the trust. The trust is a little bit difficult how to create this one. So the trust itself, it can be measured by time. When first time they do contract and then the contract is succeed, they will continue to the second time and also succeed in the third time and succeed in the fourth time and so on. So the main point here is how to do agreement, how to run the agreement well. This is the important thing. And how farm institutional model is able to solve farm problems starting from upstream to downstream. So the fairness, yeah, the fairness is the key. When they do, let's say like this, the integrator itself, they will get, I mean, the small benefit for one farmer. But because they run a lot of farmer, so the benefit, the pie is big. But for one farmer, they will get higher for one unit, and then because they size is small, so maybe they will get higher for one unit. The fairness is important. Who spend more money or more capital, they will get the higher percentage of benefit. Thank you.
Thank you very much, Rondi, for nice answer. Next question. Any other question, please?
Pak Tri, sorry, I can't hear your voice clear here.
Okay, I think we have one question again from Putri Permatasari from Universitas 11 Maret. What steps need to be taken so that farmers have high bargaining power? Maybe in this case, the role of farmer group is very important to be able to help marketing agriculture product related to quality and quantity so that they have the same standard. Halo Pak Rondi.
Mr. Rondi?
Okay, thank you. For the next question from Putri Permatasari from 11 Maret University. What step need to be taken so that farmer have high bargaining power? Maybe in this case, the role of farmer group is very important to be able to help marketing agriculture product related to quality and quantity so that they have the same standard. Okay, this is the nice question. So what step? For the first step, this is in my opinion. So my opinion is based on our research and also the previous research. So they engage in one corporation and one organization. So by engage in one organization, they will have more gain because they can produce more quantity, and then by producing more quantity, so they have a more bargain position to the integrator or to the company. So by doing so, so maybe they will have also access to get the credit. But we have to take care about this. The organization, when the organization is still small, so they engage in good condition. But after the organization is getting larger and larger, so maybe there is more interest thing. So I mean, they try to get their own interest, and therefore the organization it will break out. There are a lot of example for this. Let's say for one corporation in the small size, the engagement, the objective is very good. But since it's grower and grower, so they will break out because some member they will have own interest, and then they try to occupy the organization with their own self, and so on. We have to take care about this. So how to solve this one? So how to solve this one is they try to make the organization is belong to organization, not belong to one person. When the organization belong to one person, it will be like this. So in the small size of the organization, so engagement in one organization is we need to take care, we need to engage, and then the fairness, the open of the organization, something like this. Thank you.
Thank you very much for your answer, Pak Rondi. I think if audience wants to continue the discussion, please send email to the both of the presenters. Finally, we come to the end of this seminar. We would like to thanks to the both speaker, Pak Rondi and Professor Fahrul Hasan, for interesting presentation, and all participants in the seminar. Finally, please give applause to the speakers. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Time give to the MC. Thank you.
Thank you very much for our guest speaker for the spectacular presentation, and thank you very much to Dr. Trihandoyo for moderating this session. And ladies and gentlemen, we are now finally entering the session we have been waiting for, the awarding session. In this awarding session, we would like here 3 winners that get the entitled Best Speaker for the parallel session. I would like to begin for the first person that get the honor as the Best Speaker. Ladies and gentlemen, for the first Best Speaker, please give applause and congratulations to Insinyur Wayan Wangiana, MSc PhD from University of Mataram. Congratulations, sir. And ladies and gentlemen, for the second person that get an honor as best speaker, ladies and gentlemen, please give applause and congratulations to Muhammad Rizky from University of Jember. Congratulations, sir. And the last but not least, the third person that get an honor to be the best speaker of the parallel session. Ladies and gentlemen, please give a big applause and congratulations for Novi Haryanti SPMP from Brawijaya University. Ladies and gentlemen, we are now more closer to an end. But before we close this event, we would like here the closing statement by the Dean of Faculty of Agriculture of University of Jember, Professor Dr. Insinyur Sutriyono MP. Ladies and gentlemen, please give applause and give a warm welcome to Professor Sutriyono.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, alhamdulillah, alhamdulillah wa syukurillah. I will like to express how grateful we are to finish the 5th International Conference of Agriculture and Life Science in Faculty of Agriculture, University of Jember from November 3 to 4, 2021, with theme Accelerating Transformation in Industrial Agriculture through Science Implementation. This conference is held in collaboration with Program Implementation Unit of Islamic Development in Development Bank, University of Jember, and the Center of Excellence on Crop Industrial Technology. We are here to communicate and gather dissemination of information and research results in the field of agriculture as part of accelerating transformation in industrial agriculture through science implementation. Through conference, we shared the problem, idea, knowledge, and technology to arrange solutions that communication and discuss in ICALS 2021. This conference was attended by 319 participants with 216 presenters and 148 submitted full paper that will be potentially published in 8 proceeding scopus. The submitted full paper are divided to 6 seminar scopus, namely agronomy and plant protection, biotechnology and biomolecular, agriculture, engineering and technology, food science, and smart education for plant-based, smart social and policy and industrial agriculture, and smart business for agriculture and healthy foods. I would like the extend gratitude to all authors of conference from many university, institution, work communication, and share their research result. The editorial team to work together to extend conference. Fakultas Pertanian Universitas Jember sebagai suatu organisasi yang melaksanakan kegiatan seminar internasional yang bertajuk ICALS 2021 mengucapkan banyak terima kasih kepada Gatot Sumbogo Jati, Prof Peng Xiang, Prof Alexander Flor, Prof Tri Agus Siswoyo, Sebastian Konak, Prof Velavan, Prof Fahrul, and M. Rondi, PhD. Last, the hope that all participants of the conference have a great experience and memories, even some of you attended this meeting virtually. We hope to see you again the next conference. Stay safe and stay healthy. Jaga kesehatan, jaga kerukunan, jaga jarak, supaya kita berbahagia. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Sutriyono, for the closing remark. Ladies and gentlemen, for your additional information, the winner of the Best Speaker will get the certificate of Best Speaker of the 5th International Conference on Agriculture and Life Science 2021 from the committee. So, ladies and gentlemen, time goes so fast, and finally, this event is coming to close. We are now entering our last agenda. There is closing and documentation. So please to all participants to turn your camera. Thank you very much, ladies and gentlemen. I would like to counting down for the first slide for taking a picture together. Please let me start. 3, 2, 1. Followed by the second slide. Ladies and gentlemen, please turn on your camera. Okay, I will start. 3, 2, 1. Thank you very much, ladies and gentlemen. Finally, this is the end of the 5th International Conference on Agriculture and Life Science 2021, Accelerating Transformation in Industrial Agriculture Through Science Implementation. Ladies and gentlemen, let us close this event by saying hamdalah together. Alhamdulillahirabbil alamin.
Alamin.
For the last, I do apologize if there was a mistake. Thank you very much for your kind attention, and see you again in the next International Conference on Agriculture and Life Science 2022. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
See you again next year.
Waalaikumsalam warahmatullahi wabarakatuh.
See you next time.
monggo yang apa foto di depan sini untuk dokumentasi panitia harus pakai masker. Monggo Pak Kaprodi, Pak Cahyo, monggo Pak Cahyo, Pak Kacung, monggo di atas saja, cukup enggak?
Setiap saat, setiap waktu, keringat basahi tubuh. Ini saat yang ku tunggu. Hari ini ku buktikan, ku yakin aku kan menang. Hari ini kan dikenang, semua doaku panjatkan. Sejarahku
Matur nuwun rekan-rekan sekalian, monggo Pak Didik nanti direncanakan untuk supaya badan tidak lumrah, mungkin hari Senin setelah upacara itu supaya ada jenang sungsum begitu. Hari Senin habis upacara.